Good evening, everyone. We are Team RITALIN, research and testing ADHD's linked to impulsivity in neuroscience. My name is Sen. This is Brooke, Dara, and Taylor. Our research focuses on ADHD animal modeling under the supervision of our mentor, Dr. Matthew Rash. Okay, so attention deficit hyperactivity disorder, or ADHD, is a disorder primarily characterized by impulsivity or hyperactivity, which can influence one's ability to focus on the task or regulate one's behavior. According to the statistical data from the Centers for Disease Control, this disorder affects 5 to 10% of all school-age children, and there has been a 20-fold increase in prescription of ADHD drug in the past 30 years. As of now, there is not much well-known or experimentally verified neurological basis of this disorder. And diagnoses are mainly based on behavioral observations rather than quantitative measures. As a result, there has been frequent misdiagnosis and rising medical costs. So, one way to study ADHD is to look at prenatal nicotine exposure, or PNE, subjects. In this approach, we look at the offsprings of the mothers who smoke during pregnancy. From now on, I'm going to refer prenatal nicotine exposure as PNE throughout the presentation. So, past studies indicate that PNE is linked to many psychiatric disorders, such as drug abuse and conduct disorder. Women who smoke during pregnancy are 3 times as likely to have ADHD children, but in the United States, 1 in 5 women still smoke during pregnancy. So there seems to be a link between PNE and ADHD symptoms. So the researchers wanted to know how the rats would contribute to this kind of ADHD behavior if they were exposed to nicotine. Interestingly, they defined behavioral, neuroanatomical, neurochemical disturbances which were shown after PNE in the rats, which were very similar to the symptoms shown by the ADHD patients. Also, the application of methylphenidate, which is active agent in ADHD drug, was found to significantly decrease impulsivity in PNE rats. And PNE rats in humans with ADHD have similar deficits on behavior tests that measure impulsivity. So, because they have similar symptoms shown by the ADHD patients and PNE rats, we can assume that the PNE rats can serve as an ideal animal model for ADHD study. So as I mentioned before, PNE rats and humans with ADHD exhibit similar behavioral symptoms, which are inattention, hyperactivity, and impulsivity. Inattention is shown by difficulty concentrating, distractibility, and problems completing tasks. Hyperactivity is shown by high recessive motions, and impulsivity is signified by tendency toward rapid, unplanned actions, without considering any negative consequences of these actions. In our study, we focus on the impulsivity behavior factor of this disorder, since methylphenidate was found to decrease impulsivity in both PNE rats and human subjects with ADHD. Also, impulsivity is the main behavioral factor that determines ADHD. Okay, so how can we measure impulsivity in human subjects? We can run a stop signal task, which can measure the inhibition of an action that is already initiated. This means that this task can measure the ability to stop. So during the task, the human subjects are asked to respond as quickly as possible to the go signal, which is indicated by right or left dot in the middle of the box. And throughout the task, there will be a presentation of brief stop signals. In this case, is only 33% of the child. And this stop signal is indicated by the dot in the middle of the box. And when the stop signal is presented, the subjects must stop responding and cancel their initiated action. ADHD patients show really poor performance on this task because of their impulsivity, which makes them challenging to stop their initiated action. So as I mentioned before, PNE results in brain abnormality. And medial prefrontal cortex, or MPFC, is one of the main brain areas that is hugely impacted by PNE. MPFC is shown to be activated during the impulsivity task, and as you can see, rat has homologous brain area to the human MPFC brain MPFC in both rats and humans serve as an executive center, which is important for decision making, learning, and memory. Since rats have similar structure to the human MPFC, we can measure impulsivity by looking at the neural activity near this MPFC area in candy rats. Okay, so we wanted to learn more about PNE, impulsivity, and ADHD, and we reviewed a number of literature, and we found out that some of the aspects of the relationship between MPFC and impulsivity were not fully yet addressed or studied in any other ADHD studies, and one of which includes a full understanding of MPFC neurosignaling, which is essential to develop an effective treatment for ADHD. An experimental system with animal modeling of impulsivity, which will elucidate biological foundational behavior. And lastly, a solid correlation between behavior and neurofiring, which will allow us to pinpoint the signals involving impulsive behavior. So by looking at these areas that are not yet fully explored in any other ADHD studies, we came up with a novel study which measures single recording of the MPFC neuron of PNE rats during the stop signal task and derived the relationship between neurofiring and the behavior of this disorder. So the hypothesis of our study was that PNE rat model is a valid model for the study of ADHD-like symptoms. In our study, our main goal was to first show that PNE rats were more impulsive during performance on the stop signal task. Second, to demonstrate the activity in MPFC is correlated with performance on the stop signal task. And lastly, to evaluate neural signals in MPFC in PNE rats during performance on the stop signal task.
All right, so as for our methodology, we wanted to see the effect of prenatal nicotine exposure on impulsivity. So we began with 10 mothers, 5 of which were designated to a control group, and the remaining 5 to the nicotine group. The nicotine mothers received the nicotine through their water. We gradually acclimated our rats to this nicotine dose by beginning with the dose of 0.2 milligrams per milliliter, and over a course of 4 weeks, increasing it to 0.6 milligrams per milliliter. We settled on this final dose because through our literature review, we found that this dose was high enough to cause behavioral differences in the offspring, yet still low enough to prevent any serious developmental delays. After this, we had attained 17 male PNE pups and 23 male control pups. We were interested in males in our study because ADHD is more prevalent in males than females. Finally, we cross-fostered all of our pups to control mothers in order to control for any parenting effects, such as the nicotine pups continue to receive nicotine through their mother's breast milk. So prior to training, we wanted to determine whether there were any significant differences on several metrics. We found that there was no significant difference on the pregnancy duration, the number of pups per litter, or pup birth weight. We also, on day 30, had all of our pups complete a locomotive task, in which we found there was no significant difference on hyperactivity, another known symptom of ADHD. After determining there were no significant differences on these metrics, we then randomly selected 8 male pups from both the PNE and control groups and proceeded on with training. So training consisted of a 2-hour-a-day period during which the rats would complete approximately 150 trials. During these trials, we wanted to motivate them, and thus they were mildly water deprived because upon completing the task correctly, they would receive a small dose of sugar water. After training for several weeks, we then proceeded to perform surgery on the rats in order to implant the electrodes as the one shown here. So here at the top is a cannula. Within this cannula are 10 small wires. These wires enabled us to record neural activity of neurons within the MPFC. After surgery, we allowed our rats to return to their post-operative levels after recovery, and then we were able to continue with our training. So I'll now review the stop signal task. For our task, the task begins with the illumination of a house light within the chamber. This will then indicate to the rat to place their nose into a central port. After 1,000 millisecond delay, a directional cue light will come on either to the right or to the left. Then, on 80% of our trials, which were known as our GO trials, The rat would move in the indicated direction and receive a reward from the fluid well below, which was the sugared water. However, on the remaining 20% of our trials, which were our stop trials, the task would begin the same. However, after the first directional cue light, a second cue light in the opposite direction would illuminate. This would force the rat to stop its first initiated behavior and move in the opposite direction in order to receive a reward underneath the second light that it illuminated. Now, throughout our study, we maintained an 80-20 ratio of go to stop trials because this ensured that the go trials would be more habitual and that the rats would not become accustomed to our stop trials. So now to review our trial types. We had two types of go trials, go left and go right, in which the rat would receive a reward upon moving in the indicated direction. And we had two types of stop trials, stop left, go right, and stop right, go left, in which the rat would receive a reward upon moving in the second indicated direction. I'd like to point out to the audience here that from this point on in the presentation, green will represent go and red will represent stop on our following slides. So after data collection, we then want to review the behavioral results from our study. We found that for our control rats, they performed significantly better on go trials as compared to stop trials. When we looked at the nicotine rats, we saw a similar trend, and that the P&E rats performed significantly worse in stop trials as compared to go trials. However, when we compared the control rats to the P&E rats, we found that our P&E rats performed significantly worse in stop trials as compared to controls. Because the P&E rats' performance was not impacted and not impaired on the go trials, yet it was on stop trials, this suggests that our P&E rats were more impulsive as compared to controls. Next, we looked at movement time. Movement time can be described as the amount of time it took for the rat to complete the trial. Here we looked at three trial types. We looked at correct trials for both go and stop error was trials in which the rat completed a stop trial incorrectly. A taller bar is representative of a slower movement time, whereas a shorter bar is representative of a faster movement time. Here we can see that control rats had the slowest movement time on stop trials that they performed correctly. When we looked at nicotine rats, we saw a similar trend, in which they too performed the stop trials correctly the slowest. However, again, once we compared the P&E rats and control rats, we found that the P&E rats performed significantly faster across all trial types as compared to controls. So for our final analysis, we looked at the relationship between percent correct as a function of movement time. We found that there was a positive correlation between these two, as indicated here. This suggests that as the rats tended to move slower on the trial, their performance also tended to increase. We therefore call this a speed-accuracy trade-off. So now to review our behavioral findings. First, we found that the P&E rats were more impulsive, as indicated by their unimpaired performance on the go trials, yet their impaired performance on stop trials. Next, we saw that P&E rats were faster across all trial types as compared to controls. And finally, we noted a speed accuracy trade-off in both control and P&E rats, in which they performed, upon performing the trial slower, they performed significantly better. Now we can explain perhaps the P&E rats that perform faster across all of them by a recent study that found that ADHD children perform better on tasks that were uninterrupted and directed as compared to children without the disorder. And at this point, I will now pass it off to Gavin.
Thanks, Brooke. So in addition to the behavioral data, we also collected neural data. So whenever a neuron fires, there's a voltage change inside the cell and outside the cell. The electrodes that we implanted were able to pick up that voltage change from outside the cell. And this is what an example waveform of what we collected. Afterwards, we did histological analysis, where we extracted the brains, we sliced and stained them, and this was to determine that we were recording from the correct brain area. So you can see here, between these two triangles is the MPFC, and each black dot is an electrode. So you can see that all of our electrodes were in the correct gray area. Afterwards, we did data analysis using MATLAB. We found something really interesting. We found that the MPFC has two distinct populations of neurons. Neurons that fire more, so they increase their firing rate during a task, and cells that decrease their firing rate during a task. So from now on, we're going to refer to that as increasing type cells and decreasing type cells. So let's take a look at an example increasing type cell. So this is a raster plot. The rows represent different trials. The columns correspond to different time points within the trial. And each black dot you see is a neuron fire. This histogram is just a summation of all the neuron firings during that session. So you can see for GO trials that compared to baseline, where point 0 is the time point where the rat exited the odor port, so they started to initiate that movement. You can see that these increasing type cells increase their firing rate. Now what about stop trials? We found that for increasing type cells, on stop trials, these cells fired more. So they're involved in behavior inhibition. So when the rat had to stop its initiated movement and go to the other well, these increasing type cells fired more. What's interesting is that all cells in the MPFC are slightly biased towards directions, either going to the left or going to the right. So this cell here, again, this is an example cell, fires stronger for behavior going to the right. This is indicated by the stop trials on the right versus stop trials on the left. So for this cell, we can say that it fires more for stop trials and prefers the right direction. We're going to be using preferred direction and not preferred direction instead of saying left and right. So we decided to average all of our neurons. Again, the y-axis here is firing rate, the x-axis is time. And we found that the average neural firing rate was modulated by response direction. So for GO trials, you can see that the solid line here is the preferred direction, and the dashed line is the non-preferred direction. And it was similar that the preferred direction, these neurons fired more in both control and P. For stop trials, we also found that the firing rate increased. So in both control and PNE neurons. And we found that across the two control or experimental groups, control and PNE, that the PNE neurons were significantly reduced in their firing rate compared to the control neurons. We also wanted to see if behavior and these increasing type cells were somewhat related. So we plotted percent correct across all trial types and firing rate of these increasing type cells. And we found a positive correlation between the two, for both control and nicotine. So we can say that if as these increasing type cells fire more, the rats get more trials correct. So in summary, these increasing type cells are modulated by response direction. They're stronger during stop trials. They're correlated with percent correct. And they're significantly reduced in PNE neurons.
Okay, so as we had the increasing type cells, we also had decreasing type cells. So again, looking at an example neuron, we examined this raster plot here. So just to review, each horizontal row represents a trial, and along the vertical columns aligns on a time point, with time point 0 being the time from point at port exit, also known as the initiation of the task. So in the bottom here, in this histogram, it is simply the summation of each of these neural firings at the time point across these trials. So what makes these decreasing type cells is the fact that you look at the baseline before time point 0 and after time point 0, and you see that the neural activity is reduced after the task is initiated. And again, similarly to the increasing type cells, we have cells that favor the left and right direction, that will hereby be referred to as preferred and non-preferred directions. So looking at our 182 control decreasing type neurons and our 174 PNE decreasing type neurons, we see that similar to the increasing type neurons, they also had their neural activity modulated by the response direction. So here you see the preferred direction on Go trials, represented by the solid green line, and the Go non-preferred direction, represented by the dashed green line. And we see that in both control and PNE groups, there's increased neural firing rates in the preferred direction. So now examining stop trials as well as Go trials, we have stop trials represented in red and Go trials represented in green. So we found that, again, like our increasing type neurons, these decreasing type neurons also fired more strongly for stop trials as opposed to the Go trials. And this is evidence here. You can see in these plots, the red line is slightly higher than the green line. Also important to note, like our increasing type cells again, we see that in the PNE group, the neural firing rate was significantly reduced as compared to the control group. And this is present in both the baseline activity, which is before the task is initiated, and once the task is initiated. So you see here this reduced level of neural firing rate in the PNE group as opposed to the control group. And again, like our increasing type cells, we wanted to correlate the behavior measured by our stop signal task with the neural firing data. However, unlike the increasing type cells, we did not see a correlation between the percent correct, or the accuracy on the task, and with the neural firing rate in these decreasing type cells. However, comparing movement time to the firing rate, we did see a positive correlation in the control group between the movement time and the neural firing rate for these decreasing type neurons. Now it's important to note that although this may seem like the rats were somehow increasing their neural firing rate consciously to perform slower on the task, this is not the case. It's actually just that as the rats happen to have a greater neural firing rate on a specific task, they also happen to perform that trial slower. So to summarize this decreasing type cell data, again, like our increasing type cells, we found that neural activity was modulated by response direction. So we saw greater neural firing in the preferred direction. Neural activity was also stronger during the stop trials as it was in our increasing type cells. However, neural activity was correlated with motor output in the controls only, as opposed to with the increasing type cells, this neural activity was correlated with the percent correct. And finally, again, like our increasing type cells, we saw that neural activity was significantly reduced in the PNE neurons of rats as compared to the controls. So to bring everything together, looking at the behavioral data,